Hello, I'm Phil Ash from Grin2B Foundation, and thank you for joining our Grin2 Beehive webinar week. Our foundation understands just how daunting it can be to keep up with ongoing research regarding Grin disorders. If you're a newly diagnosed family, it can be completely overwhelming, and many just don't know where to start. To that end, we are pleased to offer this Genetics 101 presentation courtesy of Ms. Rebecca Smith of the Simons Searchlight organization. In addition to being a researcher for Simons, Ms. Smith also serves as a clinician in pediatric neurodevelopment at the Autism and Developmental Medicine Institute at Geisinger in Pennsylvania. At Simons, Rebecca works closely with participant families to answer any questions and to help make meaning out of genetic testing results. She's with us today to offer a presentation regarding the basics of genetics and how Grin2B fits into our larger genetic makeup.
Hello. Well, welcome to our talk today, where we're diving into some of the genetics about Grin2B. I know that often the genetics can seem like a whole new language when you're learning about a new diagnosis. So I hope that today we can walk through some of the genetic basics, but then also apply those to how you read a lab report. I hope that this presentation helps just make a little bit more sense and maybe some meaning out of those genetic test results as well. I'll be sure to answer some questions that we may have at the end of today's talk, and I'll also be sure to include my email at the end of the presentation so that if you have any other follow-up questions, you can feel free to contact me directly. Let's go ahead and dive on in. So I'll begin by going over some of the basics of genetics. I'd like to build a really strong foundation of when I use these words, what exactly do they mean? And then after we can establish all of those definitions, we'll move ahead to apply it to a lab report. So let's get started. When we talk about genetic testing, the first thing we think about is our DNA. Our DNA is our body's instruction booklet. It tells our body how to grow, develop, look, learn, essentially how to work. Thinking about all of our DNA, sections of our DNA are called genes. And these sets of instructions have to do with different things for our body. So one gene may be involved in brain development, where another gene may be involved in keeping our heart working and beating like we would typically expect. Our DNA is then wound up and put into packages that we call chromosomes, which we'll get to in just a second, so hold on to that. And then our chromosomes go into each of our cells, and our cells are the building blocks of our bodies. Our bodies are made up of billions and billions of cells. So thinking back to these chromosomes, the packages of our DNA, we typically have 46 chromosomes. If we would look at them under a microscope, we would sort and arrange them from tallest to smallest. And typically, we have all the same 22 pairs. Within each pair, we get one from our mother and one from our father. The last two chromosomes that we have are our sex chromosomes. So for females at birth, we would have two X chromosomes, whereas males would have an X and a Y chromosome. When we're thinking about Grin2B specifically, that gene is packaged into one of our chromosome 12s. So now let's zoom into those chromosomes, thinking about our DNA. Our DNA is spelled out in letters, the exact same way that words in a book are. The letters of our DNA are supposed to be in a very specific order for our body to know and understand how to use those instructions. And we know what the order of the DNA is supposed to be. Our body uses those DNA instructions to make proteins. How exactly does it do that? Well, it reads those DNA letters in chunks of 3. Each 3-letter code codes for what we call an amino acid. In this picture, amino acids would be each of these circles. All of these tie together to ultimately make a protein. And proteins have many different roles in the body. They're essentially what's making our body work. They're what makes our eyes see, our ears hear, our brain learn. And so we go from the letters of our DNA to proteins. Now, I mentioned that the letters of our DNA are typically in a very specific order. But what can happen is some of those letters can be changed, kind of like a typo or a misspelling. And if that happens, it can change the way that our body reads and uses those instructions. That ultimately can result in things like medical and developmental differences. So when we do genetic testing, that's what we're looking for, any changes in the spelling of our DNA. Now, it's important to remember that we all have some spelling changes in our DNA. If we did genetic testing for anybody at the grocery store, for example, we'd find some genetic changes. But not all of them affect our health and development. Some of them just make us unique and don't change anything. So that's really where your genetics providers are working now, is to figure out which changes do cause differences to somebody's health and which changes don't. So now I'd like to shift gears and talk about the different types of genetic changes that we can see. The first type of genetic change that I'll talk about is called a copy number variant. Copy number variants are changes in the amount of somebody's DNA. So a section of their DNA may be copied an extra time, and we may have more than typical. We call those duplications. On the other hand, a section of DNA may be deleted or missing. These are what we call deletions. So looking in this picture that I have over here on the right, if we think about going back to our DNA being our body's instruction booklets, a deletion would be if some of those books are missing and are no longer there for our body to read. A duplication would be like if a section of these books are copied and repeated again. So we have twice as many as we typically would in that section. These are important because the changes in the amount of somebody's DNA can also change the way that our body is using those instructions. And sometimes these may result in things like medical and developmental differences. Thinking about GRIN2B specifically, we know that this gene is located on chromosome 12. So looking at the pictures of our chromosomes that we have over here, GRIN2B, the gene, is packaged in chromosome 12 up towards the top here. So somebody may have a chromosome 12 deletion that includes the GRIN2B gene. Let's think about a different type of genetic change that we can find. Instead of copy number variants, now I want to think about sequence level variants. These are essentially if we would do a spell check on those letters of the DNA. So instead of looking for big, extra, or missing pieces, we're now looking for any individual spelling changes in the letters of the DNA. There's a couple different types of these sequence level variants that we can see. The first type that I'll talk about today is one that we call a missense variant. This is when there's a single letter change in the DNA that changes one amino acid. So let's break this down a little bit farther. Some background information I want to give to you before we dive in too deep is the difference in the different ways that you'll see these genetic changes written on a lab report. If you see a C dot, that's telling us we're thinking about the letters of the DNA themselves. So for example, in this case, this is telling me that the 404th DNA letter of the GRIN2B gene is typically a letter C, but in this case, it's been changed to the letter G. So up here, we would see typically this is a C, now it's a G. You may remember that we use chunks of 3 letters to make our different amino acids. And what some spelling changes do is it just changes this one building block coded for by these 3 letters. What that looks like in our total protein is the rest of the protein is unchanged. The other amino acids are coded for exactly the same way, but this one is different. Sometimes these types of changes affect how that protein ultimately looks and works, but sometimes it doesn't. So that's something that we use genetic research to help us understand. If you see a P dot, this is telling us that that's at the protein level. So in this case, this is telling me that at the 135th amino acid down the line, what used to be an alanine is now changed to a glycine. So the DNA letter changed, caused an amino acid change in our protein. These are missense variants. There's another type of sequence level change that we can see called nonsense variants. They start the same way by changing a single letter of the DNA. So again, to review, in this case, the 3,301st letter of the GRIN2B gene is typically a G, but in this case, it changes to a T. What this does is it doesn't change this amino acid coded for by these 3 letters. Instead, it stops the protein. In this case, none of these amino acids that come after it are red. Our protein stops there. This causes our final protein to be much shorter than we would expect, and oftentimes these proteins do not work like we would expect. Because of that, nonsense genetic changes very often related to medical and developmental differences when they occur in GRIN2B. Some ways that you would know that a variant is a nonsense variant is looking at our protein naming. So instead of having a different amino acid listed over here, you can see things like TER, meaning protein is terminated at that point. We also can show this by having a star or an X. All of these things tell us that that protein isn't made beyond that point. There's one last type of change I'd like to talk about today, so thank you for sticking with me. This is called a frame shift type of variant, and it has to do with insertions or deletions. So instead of a DNA letter being changed or substituted, it's possible that a letter of our DNA could be copied an extra time or just taken out. So in this case, what we're seeing is this letter C up here is being copied an extra time. So we have one C originally, but what happens with this variant is we have 2 C's down here. Now, this is important in thinking about those clusters of 3 letters that ultimately make our proteins. Because what these insertions or deletions do is they shift those 3-letter groups. So while at the top, the next groups of letters were ACC and then TTC, you can see that our groups down here make different letters. This means that those amino acids it codes for are different, but also that that protein doesn't look like we would expect it to. Much like nonsense variants, these proteins look very different and often are much shorter and stop much farther before we would typically expect. You can find these in the names of genetic changes because we typically see an FS at the end, standing for frame shift, and then an X telling us that that protein is ending before we would typically expect. Now, I know that this was quite a lot of information to take in all at once. I hope that potentially you recognize the type of genetic change that your child has. So I'll let you soak some of this in, and I'll now shift gears to talk a little bit about the different types of genetic testing that we can see. Some clinicians start genetic testing with a test that we call a karyotype. This is a very zoomed out view looking at the numbers of chromosomes somebody has. This will look like the big picture we saw at the beginning of the presentation, where we had all of our pairs of chromosomes sorted from biggest to smallest. The next test that many people go to is called a microarray. This test is zooming in on those chromosomes, looking for any missing or extra pieces, those copy number variants that we talked about. After that, we may move on to sequencing. Sequencing is essentially the spell check type of testing that we talked about, reading through those individual letters. Within sequencing, there's a couple different tests that we can use. The first one is a gene panel. So that's looking at very specific genes we know are related to a specific clinical indication. So for example, you can order a gene panel that's reading through the letters of genes related to things like developmental differences or seizures. Other clinicians may use a broader test like whole exome sequencing. This test is reading through all of the letters of our DNA that are telling our body how to work. This is a more comprehensive test. When we get results back, what do we make of them? This is where we get the variant interpretations. Now, I want to remind you what I talked about a bit earlier, where we said that genetic changes, we all have them. Not all of them affect our health, but some of them do. These variant interpretations are the labels that we put on genetic variants to try to say if they're affecting somebody's body or if they're not. On your lab reports, you'll likely see one of two classifications, either these uncertain variants, which we sometimes label VUS, standing for variant of uncertain significance, or you may see likely pathogenic or pathogenic. Starting with these pathogenic gene changes, these are variants that we have enough evidence to be confident that that gene change is likely a good explanation for somebody's medical and developmental differences. This means that we found our genetic answer. Sometimes, though, we just don't have enough research to be that confident yet. Maybe we haven't seen anybody else with that very specific gene change. Or maybe we see that gene change in people who don't show us any medical or developmental differences. In those cases, it may fall into this uncertain category, meaning we need more research before we can definitely tell what this gene is doing. We'll learn more and more about genetics every single day, so uncertain variants don't always stay uncertain. It's possible that as we learn more in the future, we're able to upgrade to pathogenic or downgrade to benign. You probably wouldn't see these benign or likely benign variants on genetic testing. That's because these are gene changes that we don't think are affecting somebody's health and development. Because they're not playing a role for somebody, we often don't report them on our lab reports. To finish up our Genetics 101 session, I'd like to call out some specific terminology and just go over some of this vocabulary for things that we'll see on lab reports or hear during genetic talks. The first one is autosomal dominant. This idea goes back to the 2 copies of each gene that we get, one from our mother and one from our father. An autosomal dominant condition means that if somebody has a genetic change in one of their 2 copies, that's enough for them to show medical or developmental differences. Some conditions, you need genetic changes on both copies. Those are what we call autosomal recessive conditions. But for GRIN2B, we're following an autosomal dominant pattern, meaning just one change is what we see. When a person has a pathogenic genetic change in GRIN2B, they're called heterozygous. That means that of their 2 copies of the gene, one of them has a genetic variant in it. We also think about, well, where did this genetic change come from? And there are 2 possibilities that we consider. The first would be that the genetic change was passed down or inherited from a parent. In that case, if we tested the child and that parent, we would see the same genetic change in both individuals. It is very important to remember that we cannot control our DNA, if our DNA has any spelling changes in it, or what DNA we pass on to our children. This is a totally random. The other possibility is that the genetic change may be brand new, only in the child. This would mean that both parents test negative for the gene change. If it's a new change, only in the child, that's what we call de novo. It's very important to remember as well that absolutely nothing that anybody does or doesn't do causes these random gene change. They're totally by chance events that occur randomly. One last topic to review before we move into reviewing a lab report. And this is something that you may see on a report or you may not. To differentiate what we're seeing, I first want to go back and explain that typically, when we think about a genetic change like a GRIN2B diagnosis, that means that that GRIN2B gene change is in every cell of somebody's body. It was likely present either in the egg or the sperm prior to conception. So after the egg and sperm met, every cell down the line that was made has that gene change. What we can sometimes see, though, is that people can also be mosaic for a gene change. So that's what this picture is showing here, that the red cells throughout somebody's body has, in this case, a GRIN2B gene change. This would happen if at some point during development, the change happened new in a cell. And at that point, any cells that come from that one would have the gene change, while their other cells would not. Again, this is what we call mosaicism. These individuals who are mosaic may show us clinical signs, but they may not as well. It really just depends on what parts of their body have the specific genetic change. This is different from something else you may read about called germline mosaicism. Germline mosaicism is when some of a woman's eggs or some of a man's sperm carries the specific gene change, while the rest of their body does not. Individuals who have germline mosaicism would not show us any medical or developmental differences. But that does mean that multiple of their children may be born with one of these genetic changes, even if they themselves test negative. This is rare. We think this happens less than 1% of the time, but it is something that we have seen before, and so is important to share with our families. Alrighty, we have made it through the brunt of the genetics, so I will let your brains take a minute to soak that all in. I'll grab a sip of water, and then we'll dive into a lab report to think about how we can connect this all. So this is an example of a GRIN2B lab report that we can get back. Lab reports from different genetic testing laboratories may look a little bit different, but they're all made from the same parts and pieces. So even if it's in a different order, I hope that you'll be able to find the corresponding sections. The black boxes I included here just to take out that patient information, so we're not sharing any medical histories. We'll, of course, start at the top. At the top, you'll see all of the demographic information for your child, and you'll also see the date of testing. This is important, thinking about how much we're learning every single day. If your testing is maybe 2 years old or older, I encourage you to get that testing updated. You can ask your clinical provider if there's any new information, and they may be able to either look up the new information themselves or send it back to the genetic testing laboratory to see if they know of any new information. It is a great thing, and I definitely encourage you to continuously ask your providers if there's any new information. Moving down that test report just a little bit, you'll see the type of testing that was done. Like we talked about before, this could be a microarray, looking for those extra or missing pieces, or it could be some of the sequencing testing that's doing the spell check on the DNA. Moving even further down, you'll see a section that's labeled clinical indication. This also may be called relevant or pertinent information as well. This is really why the person's getting genetic testing. It's probably a pretty brief history of your child's medical and developmental histories as well. And then we really get into the meat of the results. There's a lot of important pieces here, so we'll take it step by step together. The first thing that you'll see is the gene that we found a change in. In this case, we're thinking about GRIN2B. This is an important first step to think about how this gene is working in somebody's body. What is its role? The next thing we think about is, well, what happens if somebody has a pathogenic variant in that gene? Most oftentimes at the top section of the report, you'll see these things listed. So in this case, GRIN2B and GRIN2B related disorder. And farther down the report, they'll give you more details and more information about, well, what are those features that we can see? Moving ahead though now, there's typically a section labeled mode or type of inheritance. And that's getting back at that autosomal dominant pattern that we talked about, where somebody has a change in one of their 2 GRIN2B copies, and that's enough to show medical or developmental differences. Moving ahead to the variant type. This is the name of the genetic change. This tells your provider exactly what DNA is changed within the gene. Thinking back, this C dot is telling us the very specific DNA letter that's changed, and the P dot is telling us the protein difference that happens. Zygosity reminds us of the heterozygous term that we learned, relating directly back to that autosomal dominant. Heterozygous is telling us that somebody has a genetic change on one of their two copies. Looking further on the report, we can see the inheritance. So it's possible that if we test parents, we can find that either an individual's mother or father has the same gene change. That would be when it's inherited. Or it could be a brand new change only in the child. And that's what we would call de novo. This is important for many families because it can help us learn the chance for other family members or future children to also have the same gene change. That's something that your clinical provider would be able to clarify for you based off of your genetic testing report. And then lastly, you'll find the classification or interpretation of the genetic variant. This is again how confident we are that this specific gene change is somebody's answer or explanation for their developmental history. This can again change over time as we learn new information and have more research. As I discussed then, at the bottom, you'll typically see a blurb of information that tells us about the gene itself and the different signs and symptoms that we can see when somebody has genetic changes in that gene. This is something that your clinical provider should also talk with your family about when you meet to discuss these results. And that brings us through a lab report. I know there are many parts and pieces, but I hope that today going over what these words mean and how we put them together to make a full picture for somebody helps give you a little bit more meaning. I'm happy to talk about any questions that we have now, but also I would be more than happy to review any of your individual lab reports or answer any questions you have after watching this presentation. I've included our email here at the Simon Searchlight so that you can send anything our way and we'll get back to you as soon as we can. Thanks for tuning in.
Thank you very much, Ms. Smith, for the presentation. We really appreciate it. We do have a few questions to ask. So the first question is, in regard to the different variants you discussed, whether it's duplications, deletions, missense, nonsense, all of those different potential types of variants, is there a variant considered to be more or less severe than others, or does it ultimately matter?
That's a great question. And so today we talked about the first step of understanding how a variant is changing the DNA. The step going beyond that is to say, well, how does that affect how the protein's working? And so that really gets at the question of, well, how will that affect somebody's health or development? And so if we have a change that's breaking the DNA, not letting the protein work like we would expect, that's when we're seeing the developmental and medical differences that we primarily think about. And so there are some types of genetic changes, like those nonsense and frame shift variants, that we know are more likely to break the gene, have what we call a loss of function variant. But those missense variants can do the same thing. So it's certainly a stepwise function where we think about all of those different pieces of the puzzle to get that final answer of whether or not this gene change is a good explanation for somebody's history.
Thank you. And then I know another popular question, as our community of GRIN2B patients increases, there is always sort of a desire. I know my family has certainly been kind of part of this, or at least we were initially, to find a quote unquote genetic twin. In other words, another GRIN2B patient that has the exact same variant in the exact same location on their genetic report. And so when you find this individual, though, least and I'm only going to speak from my own personal experience, we found that I guess it's nice to know, but it doesn't necessarily mean that the symptoms and the phenotypes that are associated with whether it's my daughter and this other patient, they don't show the same, even though the genetic variant is the same. And so the question I want to ask is, why would that be?
Yeah, that's another great question. So when we find a genetic change like GRIN2B in this case, we know that that's a piece of somebody's puzzle. It can be a large piece of their puzzle, but we know that there are other factors contributing as well. We have about 20,000 genes in our body. So even if this is how one gene is working, we still have all of those other genes that are affecting our health and development. We can think about environmental factors as well. So we know that there are many, many contributions that are coming in to eventually make somebody's full picture for what we see when we think about their health history. And so that's why even if somebody has the same exact genetic change, a lot of those other factors aren't the same between those two people.
Yeah, that makes sense. Thank you very much for answering that. This next question, I'm going to ask you to play an insurance agent. I don't know if that's something you're comfortable with. Sure. The good news is I feel like the landscape has changed quite a bit, even in the years since my own family's journey to a diagnosis. But the bottom line is that the genetic testing that is discussed in your presentation, be it whole exome sequencing or microarray, et cetera, isn't always covered by insurance companies. And so what can we as individuals do to pressure companies to cover this?
Mm-hmm. That's also a wonderful question and something that our whole field is trying to improve. I think as more people have access to genetic testing, that's just arming us with even more research supporting us to say this testing is important. That's what we as clinicians use to give insurance companies to say this testing does matter and this is why. So we're seeing that as those additional publications come out, as we're able to give those research reports to insurance companies, testing for coverage for testing is increasing. It's certainly not moving as fast as we would all hope, but I am hopeful that in the next couple of years, access to testing will be much more broad.
Thank you. One final question. So one of the hot topics in our community as we sort of move on is the idea of gene therapy. And we know that gene therapy deals with changing someone's genetic code by inserting a corrected genetic code into someone. Are you able to explain a little bit with how that works? So I guess some of the questions that are associated with that generalized topic is, how is the genetic code changed in each of our cells through gene therapy? Or is this something that would cause patients to sort of have mosaicism, as you talked about, where maybe some of the changes show up on some of the cells, but not all of them?
Mm-hmm. So that's a wonderful question and really exciting to think about what the future could hold in terms of gene therapies. I don't think I have a good umbrella answer for that in terms of what that would look like for GRIN2B specifically. We're really on the cutting edge right now of gene therapy for a few very specific conditions. And so I think that's something that over the coming years, we'll have a much clearer answer for in terms of what types of gene therapies are most effective, because they work slightly differently. And depending on the type that we're using, that would change the answer to the questions that you asked. So I don't think we're at a point right now to have very clear answers about those things, but that's something that I would definitely encourage your group to check in on a yearly basis, because I think our answers will get more and more clear as time goes on.
Ms. Smith, thank you very much for your time. Really appreciate it. And viewers of this, if you have specific questions, please refer back to Ms. Smith's email. You can also contact GRIN2B Foundation. We can forward questions to Ms. Smith as well. So thank you very much. We really appreciate it.
Absolutely. Thanks so much for having me.